Alright, Exam 1 Study Guide. Organization of life. What is biology? It is the scientific study of living organisms, both living and dead. Define. Cell is the basic unit of life found in all living organisms. An organism is a single living individual. 1 second. There we go. Characteristics of living things. Number 1, they are organized. Number 2, they use energy. Number 3, maintain internal constancy, also known as homeostasis. Number 4, they reproduce, grow, and develop. And number 5, evolve. Examples of homeostasis would be shivering to maintain body temperature or regulating nutrition with water balance. Level of organization, it goes atom, molecule, organelle, cell, tissue, organ, organ system, organism, population, community, ecosystem, biosphere. One more time, that is atom, molecule, organelle, cell, tissue, organ, organ system, organism, population, community, ecosystem, biosphere. Prokaryotes versus eukaryotes. Prokaryotes are single-celled, have no nucleus or organelles, and eukaryotes contain nucleus and organelles and can be single or multi-celled. Examples are things like animals or plants. Taxonomic groups. The most inclusive is domain. The least inclusive is species. 3 domains are bacteria, archaea, and eukarya. And 4 kingdoms are in domain eukarya are Protista, Plantae, Fungi, and Animalia. One more time, that is Protista, Plantae, Fungi, Animalia. Timeline of life. Earth is approximately 4.6 billion years old. First life appeared 4 billion years ago. Photosynthetic organisms came around about 2.5 billion years ago. Land, plants, and animals about 0.5 billion years ago. And humans about 500k years ago. Atmosphere change. Photosynthesis introduced oxygen, forming the ozone layer and enabling organisms to move onto land. The unifying principle of biology is evolution. Alright, evolution is change in genetic makeup of population over time. Artificial selection is selective breeding by humans. Natural selection is survival and reproduction of individuals with favorable traits. Adaptations are traits that enhance survival or reproduction. Scientific method has 5 steps: observation, question, hypothesis, experiment, and conclusion. Dependent variable is the measured outcome. The independent variable is the factor changed by the researcher. Standardized variable is held constant across all treatments, and the control is the baseline for comparison. Differences between atoms, elements, and compounds. An atom is the smallest unit of an element. An element is a pure substance, which is one type of atom. A compound is a molecule with 2 or more different elements. Particles of we got the particles of an atom: the proton, neutron, and electron. You already know what these are: positive charge, your negative charge, your neutral charge. Got some definitions. Atomic number is the number of protons. Atomic weight is the average mass of all isotopes of that atom. The mass number is the protons plus the neutrons. And the isotope is the same element, different neutrons. Chemical bonds. You got some types of these, right? You got ionic transfer of electrons, which forms ions. Example is NaCl, which I believe is salt. Covalent, which shares electrons, can be polar or nonpolar. hydrogen, which are weak bonds between molecules, like water, and van der Waals forces, which are weak temporary attractions. Redox reactions involve two things: oxidation and reduction. Oxidation is the loss of electrons, and reduction is the gain of electrons. The oxidizing agent is reduced, and the reducing agent is oxidized. Now for water, there are two bond types. Within the molecule, there's polar covalent, and between molecules are hydrogen bonds. Characteristics we got cohesion. Water molecules stick together. Adhesion. Water molecules stick to other substances, and less dense as solid. Ice floats due to hydrogen bonding. Acids and bases. Acid denotes H+ ions. Example is HCl. Base accepts H+ ions. Example is NaOH. And the pH scale measures H+. Lower is acidic, and higher is basic, and 7 is the neutral. A buffer stabilizes pH. An example is carbonic acid, or example is carbonic acid bicarbonate system in blood. And governed by law of mass action, reaction shifts to minimize changes in pH. There you go. That is Exam Study Guide 1. Biology Exam 2 Study Guide. Acids are molecules that dissolve either partially or fully in water and give up a hydrogen. Bases accept hydrogen when in a solution. Functional groups are groups of atoms that occur within organic molecules and confer specific chemical properties to those molecules. The pH scale goes from 0 to 14 and is determined by the equation pH equals negative log of H+, where H+ equals the molar concentration of H+ ions. 7 is the neutral of the pH scale. Above 7 are bases, below 7 are acids. A buffer, is a weak acid and corresponding base, which help to maintain pH. Buffers protect against sudden changes in pH. The law of mass action states that rate of a reaction is proportional to the product of the concentration of each reactant. Isomers are molecules with the same formula but different shapes. These examples of these isomers include structural isomers, which differ in how the atoms are joined. Cis-trans isomers have different orientations around double bonds, and optical isomers are mirror image versions of the original molecules. Organic compounds consist of one or more carbon atoms covalently linked to atoms of other elements, most commonly nitrogen, hydrogen, and oxygen. There are 4 major types of functional groups. Hydroxyl, which forms hydrogen bonds with water and has a formula of OH. Carboxyl, a weak acid that attaches to larger molecules and has a formula of COOH. Amino can receive a hydrogen, has a formula of NH2+ or NH3+, and phosphate can change the shape of a protein and has a formula of PO4+. Organic compounds all consist of monomers, which are smaller building blocks of each, combined together. 4 types of organic compounds. First up is proteins. The monomers of proteins are amino acids. The general structure of an amino acid consists of a carbon attached to a carboxyl group, an amino, an R group, and a hydrogen. They are bonded together with peptide bonds. When amino acids are connected with the peptide bonds, they form polypeptides, which are formed as linear chains of amino acids. There are 4 different structures of amino acid. The primary structure is just the base sequence of amino acids, but it actually determines all the other structures. The secondary structure refers to the localized regularities within the overall structure. Examples of this is how alpha helixes and beta pleated sheets exist within the amino acid structure. The tertiary structure is how the whole polypeptide folds, and the quaternary structure is the overall shape of a protein with interaction between multiple polypeptides. Not every polypeptide has a quaternary structure. Denaturation messes with the state of the polypeptide and is caused by things such as temperature change and change in pH. However, as long as they still have the primary structure, the proteins can refold themselves into the correct shape. Chaperone proteins protect them while they do this. Proteins have a variety of functions which are determined by their shape, including forming bone, tissue, or muscle. Nucleic acids. The monomers of nucleic acids are nucleotides. These nucleotides all chain together to create nucleic acids. The two different types of nucleotides are purines and pyrimidines. Purines always bond to pyrimidines. Sure. The purines are adenine and guanine. The pyrimidines are cytosine and thymine, which is replaced with uracil and RNA. DNA has deoxyribose, while RNA has ribose. RNA is single-stranded, but DNA is double-stranded. Nucleic acids are added 5' to 3'. Replication is the way DNA is reproduced. Transcription is the process of producing a strand of RNA from a strand of DNA. Translation is the way that messenger RNA, or mRNA for short, is decoded to produce a specific sequence of amino acids. Nucleic acids are things such as DNA and RNA, which are important as DNA codes RNA, codes proteins. They also store genetic information, which is extremely important for determining everything about an organism. Next up is lipids. Lipids do not have a traditional monomer. They have a triglycerol structure, which means they consist of 3 fatty acids bonded to a glycerol. Saturated fatty acids stay close together, while unsaturated stay far apart. Lipids are amphipathic, meaning that they can be both polar and nonpolar. Lipids are the building blocks of all living cells. What this means is they do things such as regulating hormones and storing energy in the form of body fats. And then you've got carbohydrates as the fourth one. Monosaccharides are the monomers of carbohydrates. The general formula of a carbohydrate is C1H2O1. The formula of glucose is C6H12O6. Monosaccharides are of course just one simple sugar. Disaccharides consist of 2 of those simple sugars bonded together. Oligosaccharides consist of anywhere from 3 to 20, and polysaccharides are anywhere between 20s or anything more than that 20 simple sugar mark. Carbohydrates are the body's main source of fuel. Hydrolysis breaks a polymer down into monomers, releasing energy and consuming water. Then a condensation reaction, or another word for it is a dehydration synthesis, combines monomers into polymers using energy and removing water. Spontaneous generation is a false theory that was previously held on the origin of life, claiming that living things would spontaneously generate due to certain things, such as flies appearing around rotten meat. This theory was first disputed by Francesco Redi, who constructed an experiment with 2 jars, each containing rotten meat. One jar was open and the other was covered with a cloth. Flies were found in the first jar, but not the second. Louis Pastel's experiment was similar, but used broth instead of flasks. We used broth stored in flasks, I should say, and took down the false idea once and for all. The 2 theories of how life first arose are chemical evolution, which puts forward the idea that the basic building blocks of life on Earth formed slowly over time, and the outside of Earth theory, which postulates that the first building blocks entered Earth on a meteor. The Miller-Urey experiment was an experiment attempting to prove evolution was possible by recreating the conditions scientists believe would have existed at the time. Catalysts are the key to the origin of life. Ribosomes are RNA capable of acting as enzymes. It is believed that RNA was the first large thing necessary for life that formed. Protocells were likely the first cells to form in evolution. They were much simpler and less tightly put together, and would be the stepping stone used for more complex cells. Cell theory is a major unifying principle of biology. It has 4 main tenets. Number 1, cells are the basic unit of life. They don't get any simpler than that. That's the simplest that life gets. Every organism is composed of cells would be number 2, which it's pretty straightforward what that means. Number 3, all cells come from a pre-existing cell. And numero quattro is all modern cells evolved from a common ancestor. This means that all cells have similar functions. Life is continuous, and the origin of life was the origin of cells. Eukaryotic cells are typically at least 10 times as large as prokaryotic cells. All cells have membrane, DNA, ribosomes, and cytoplasm, regardless of if they are eukaryotic or prokaryotic. The cell membrane is the outer boundary of a cell and is selectively permeable in order to let some things in and keep others out. It is a double layer formed from lipids and proteins. A phospholipid has 2 fatty acid tails and 1 fatty acid heads attached to a phosphate. What all prokaryotic cells have: 1, a cell membrane, the outer covering separating the cell's interior from the outside environment. 2, a cytoplasm, a jelly-like fluid that fills up the entire cell interior except for the organelles. All cellular structures are suspended in the cytoplasm. Number 3 is DNA, the generic material that helps in their reproduction. And number 4 is ribosomes, which help in protein synthesis. A cell membrane is selectively permeable, while if a cell had a cell wall, it is located outside the membrane and is rigid. A capsule is a gelatinous polymer made of either polysaccharides or polypeptides. They surround a cell and can help it stick to other cells or various surfaces. Flagella are appendages that help cells to swim, and pila are hair-like structures that help cells to swim. I don't think that's accurate. I think it's supposed to be helps them to stick, not swim. So I'm just gonna look that real quick. Okay. Hello, me in the future. Please do not get annoyed at this part. You were very tired. Please remember that. DNA determines everything about an organism by coding for RNA. Ribosomes are the site of protein synthesis, meaning that they work with the RNA to create proteins. The rough endoplasmic reticulum is where the RNA find those ribosomes, and they work with them to create the proteins. The smooth ER has several important functions, including the detoxification of various things and lipid synthesis. The Golgi apparatus is where the unfolded proteins go after their creation in the rough ER. It folds them, sorts them, and sends them on their way. Lysosomes can digest things taken into the cell, and peroxisomes break down hydrogen peroxide, which is H2O2. The nuclear houses the DNA and... Hold on, back it up. You're too tired. Try again. The nucleus houses the DNA and controls DNA transcription and replication. Yep. The nucleus is the largest organelle in the cell. It stores DNA on the inside and houses the nuclear envelope on the outside. It is also surrounded by the rough endoplasmic reticulum and the ribosomes that contains. The endomembrane system is an interconnected system of membrane-bound components. It includes the nuclear envelope, the two ERs, so the rough endoplasmic reticulum and the smooth endoplasmic reticulum, the Golgi apparatus, vacuoles, vesicles, lysosomes, and the cell membrane. The mitochondria is where ATP is produced. This is important as it is a key source of an organism's energy. The mitochondria has a rather unique structure. It has an outer membrane, an inner membrane, and an intermembrane space, which is located between those two, and the matrix, which is in the innermost area, and the mitochondria can only be found in eukaryotic cells. The chloroplast is a site where photosynthesis takes place. The structure of the chloroplast is actually very similar to that of the mitochondria, with it housing, of course, an outer membrane, an inner membrane, an intermembrane space, and an inner area called the stroma. Fancy schmancy. They are found in the cells of plants and photosynthetic algae. The evolution of eukaryotic cell components likely came from a symbiotic relationship between multiple prokaryotic cells. As an example, aerobic bacteria were actually engulfed into other cells as part of this symbiotic relationship, and they later, through those all being absorbed into those, became those cells mitochondria. This then led to compartmentalization, which led to these cell parts acting efficiently with one another and compartmentalizing tasks so that the cells lead to the cells we know today that are the most efficient and effective. Phospholipids have one hydrophilic fatty acid head and two hydrophobic fatty acid tails, all attached to a phosphate. The type and length of these fatty acid chains can vary, leading to different levels of fluidity in the cell membrane. The fluid mosaic model describes the structure of the biological membrane, which is constantly changing. As mentioned above, the fluidity depends on the type and length of the fatty acid chain. The cell membrane is selectively permeable, which means it can vary in what it allows to enter in order to make sure the right stuff comes in instead of the wrong stuff. Cell recognition is the process by which cells identify and communicate with each other. They utilize cell adhesion to stick to each other and their extracellular matrix. And cell junctions are specialized structures that form between cells, allowing for communication and adhesion between those cells. A tight junction. So these create a barrier that prevents the passage of substances between these cells, maintaining a selectively permeable layer, and this is common in epithelial cells. I don't think that's important. ChatGPT did though, so we're keeping it in, I guess. Adherence junctions these anchor the cells together and connect to the cytoskeleton, providing mechanical strength and stability, and a gap junction allow direct communication between adjacent cells by forming channels that enable the transfer of small molecules and ions, facilitating rapid signaling. On to the last page, you're almost there. ECM stands for the extracellular matrix, which is a complex network of proteins and carbohydrates located outside of the cell. Collagen is an example of a protein that binds to the ECM. You need to remember collagen for some reason. Just remember collagen, 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 collagen. Collagen is an example of a protein that binds to the ECM, and you better remember that or you're going to be sad. There, good song. The types of transport are active transport and passive transport. Passive goes with the concentration gradient, and active goes against it. Diffusion and facilitated diffusion are both driven by the concentration gradient, but the latter is also facilitated by proteins. Osmosis is driven by the difference in water concentrations across cell membranes. while primary active transportation is driven directly by the hydrolysis of ATP, and secondary is driven by energy stored in the concentration gradient of another molecule. Facilitated diffusion, as well as primary and secondary active transport, require transportation proteins. The active transports require energy inputs, while the pathid methods do not. The rate of diffusion can be determined, we're going with that, by number 1, the distance being covered. Number 2, the size and mass of the molecule. Number 3, the temperature of the solution. And number 4, the density. Osmosis is the diffusion of water. Hypertonic means something has a higher concentration of solute. Isotonic means places have an equal concentration solute. And hypotonic means it has a lower concentration of solute. Contrary to what is normally expected, water moves into the area of higher solute concentration, or in other words, the hypertonic location. Turgor pressure is the pressure my writing here is incredible. Is the pressure fluid inside of the central vacuole of plant cells exerts against the cell wall. So what it's trying to say basically is that turgor pressure is inside of the vacuole of plant cells. It exerts force against the cell walls because it's pressure and it pressures the walls. Makes sense. 3 proteins involved in transportation. There are types of these. We got some pumps, we got co-transporters, and we got vesicle transporters. Primary active transport moves ions or molecules across the membrane, going against the concentration gradient, utilizing transport proteins in the process. Secondary couples the movement of one molecule down its gradient with another up its gradient. Primary transportation utilizes the breakdown of ATP as an energy source, while secondary uses ions going with the gradient as a source of energy. Almost there, just a couple more things. And endocytosis is the process by which a cell engulfs its substances from its surroundings, while the exocytosis is the process of cells expelling materials into the outside environment. And then finally, phagocytosis is the process of a cell eating, and pinocytosis is the process of a cell drinking. And there we go, a 20-minute long recording. Good gosh. Alright, Exam 3 Study Guide. Metabolism is the sum total of all chemical reactions occurring in a biological system at one time. There are 2 types of metabolism. Anabolic reactions are reactions that create complex molecules from small molecules, while catabolic reactions break down complex molecules into simple ones. Anabolic reactions use energy, and catabolic reactions release energy. Energy is the capacity to do work, or the capacity for change. Chemical energy is stored in bonds. Electrical energy is the separation of charges. Heat energy is transferred due to a difference in energy. Light energy is electromagnetic radiation stored as photons. And mechanical energy is the energy of motion. The laws of thermodynamics apply to all energy transformations in the universe. The first law of thermodynamics is energy cannot be created or destroyed. The second law is that when energy is converted from one form to another, some of that energy will be made unavailable to do work. There are important distinctions between potential and kinetic energy. Potential energy is stored energy, while kinetic energy is energy in motion. Entropy is the measure of disorder in a system, which is basically energy lost as explained in the second law of thermodynamics. And enthalpy is total energy, and free energy is usable energy that can do work. Change in energy is measured in either calories or joules. A negative change in free energy means free energy is released, and a positive change in free energy means that free energy is required. And the free energy symbol for that is G, so it's plus delta G or minus delta G. Exergonic reactions release free energy and are catabolic. Endergonic reactions consume free energy and are anabolic. ATP is important because it is the main source of energy for living organisms. ATP formation is endergonic and ATP hydrolysis is exergonic. ATP hydrolysis is ATP plus water equals ADP plus phosphate group plus free energy. Two characteristics of ATP that allow the release of free energy when hydrolyzed. Number one, phosphate groups have negative charges and repel each other. The energy needed to get them close together to bond is stored in the PO bond, and then free energy of the PO bond is much higher than that of the HO bond that forms after hydrolysis. Enzymes are biological catalysts. A catalyst is something that speeds up a chemical reaction but does not make it occur if it would not otherwise occur. How they do this is by lowering the activation energy required for that reaction to occur. The activation energy is the amount of energy required to start a reaction, of course. So the transition state is the reactive mode of the reactant after sufficient energy has been supplied to start the reaction. Transition state intermediates are unstable reactants with higher free energy. Substrates are the reactants, and the active site is the location on the enzymes that the substrate binds. There are three methods of enzyme action. Number one, enzymes can orient substrates to help them react. Number two, enzymes can make the bonds unstable by stretching the substrate. And number three, enzymes can temporarily add chemical groups. There are two ways to regulate enzymes. Regulating gene expression can control the number of enzymes that are made, or the enzyme itself can be regulated through methods such as changing the shape or blocking it with regulators. Irreversible inhibition is caused by the inhibitor covalently binding to the side chain of the active site and disabling the enzyme permanently. Reversible inhibition is when the inhibitor binds non-covalently to the active site. Competitive inhibitors compete with the natural substrates for the active site. Uncompetitive inhibitors bind to the enzyme substrate complex, preventing the release of products, and non-competitive bind to a place other than the active site. Allosteric regulation is where an effector binds the enzyme at a spot other than the active site and changes its shape. The commitment step is the first step of the reaction, followed by the others in sequence. Feedback inhibition is where the final product acts as a non-competitive inhibitor that binds to the first enzyme, shutting down the reaction. Every enzyme is most productive at a particular pH, and being at a different pH can limit the effectiveness. Enzymes can also are most productive at a particular temperature and can be denatured at high temperatures. There are 5 principles of metabolic pathways. I might read through these twice just because I don't really remember these very good. Number 1, complex transformations occur in a series of separate reactions. Number 2, each reaction is catalyzed by a specific enzyme. Number 3, many metabolic pathways are similar in all organisms. Number 4, in eukaryotes, metabolic pathways are compartmentalized in special organelles. And number 5, key enzymes can be inhibited or activated to alter the rate of the pathways. Once again, starting from the top, these are the 5 principles of metabolic pathways. Number 1, complex transformations occur in a series of separate reactions. Number 2, each reaction is catalyzed by a specific enzyme. Number 3, many metabolic pathways are similar in all organisms. Number 4, in eukaryotes, metabolic pathways are compartmentalized in specific organelles. And number 5, key enzymes can be inhibited or activated to alter the rate of the pathway. Metabolism of glucose is, the chemical formula is this: C6H12O6 plus C6O2 equals 6CO2 plus 6H2O plus free energy. In other words, it is glucose plus oxygen. Is glucose plus 6 oxygen gas equals 6 carbon dioxide and 6 water plus free energy. It is a highly exergonic reaction, and it drives endergonic reactions. The 3 catabolic processes that harvest energy from glucose are glycolysis, cellular respiration, and fermentation. Redox reactions are reactions where something gets oxidized and something gets reduced. The thing that is oxidized loses electrons, and the thing that is reduced gains electrons, which reduces its overall, which gives it a negative charge, like in the reducing to a negative. Makes sense? The reducing agent gets oxidized, and the oxidizing agent gets reduced. So all of that makes perfect sense. The transfer of electrons is heavily associated with hydrogen. When hydrogen atoms are lost, that means the molecule is oxidized. NADH, FADH2, and NADPH are all electron carriers. NADH and FADH2 are used in cellular respiration, and NADPH is used in photosynthesis. What makes that easy to remember is the P, and that does not actually stand for photosynthesis, but P for photosynthesis kind of makes it easier to remember. The 5 energy-yielding metabolic pathways are glycolysis, pyruvate oxidation, the citric acid cycle, the respiratory chain, and fermentation. These occur in the mitochondria. Aerobic respiration requires oxygen to occur, while anaerobic does not. Aerobic is by far the more effective of the two. When O2 is present, glycolysis, pyruvate oxidation, citric acid cycle, and the respiratory chain are all used. When O2 is not present, only glycolysis and fermentation occur. Glycolysis is the first step of all respiration. It occurs in the cytosol and breaks down glucose into 2-pyruvate. It also produces 2 ATP and 2 NADH. Next is pyruvate oxidation, which takes place in the mitochondrial matrix. The pyruvate are oxidized as they enter the mitochondria, and in turn, NAD+ is reduced to NADH. And then what remains of each pyruvate is acetate, which binds with coenzyme A in order to form acetyl-CoA. These acetyl-CoA then enter the citric acid cycle. The citric acid cycle occurs in 3 distinct steps. In the first of these steps, oxaloacetate, a 4-carbon molecule, binds with acetyl-CoA, which yields citrate. The citrate is then rearranged and oxidized, which yields 3 NADH and 1 FADH2 and 1 ATP per turn. In the third step, the original 4 carbon molecules the original 4 carbon molecule is reformed, and the cycle begins anew. And so during the cycle, the acetyl-CoA is oxidized, and the overall yield is 4 CO2, 2 ATP, 6 NADH, and 2 FADH2. At this point in aerobic respiration, just 4 ATP have been produced. However, 10 NADH and 2 FADH2 have been formed, which are, of course, the electron carriers. These electron carriers are then used in oxidative phosphorylation. This phase occurs in 2 steps: electron transport and chemiosmosis. The electron carriers travel through the transport chain, donating electrons as they go. The energy from these electrons is used to push protons into the intermembrane space of the mitochondria. The final electron acceptor of this chain is oxygen, which combines with hydrogen to form water. And then, in chemiosmosis, the protons move down the concentration gradient from the intermembrane space to the matrix, moving through ATP synthase, producing ATP. 2 ATP are formed in glycolysis, 2 are formed in the citric acid cycle, and 28 are produced in the electron transport chain. So in total, 32 ATP are formed. Everything listed here after glycolysis is considered aerobic. Oxygen isn't directly used before the transport chain, but without it, the other steps get backed up. This is because electrons can't be donated, meaning NAD plus and FAD won't be produced. The proton motive force is generated across a membrane and has two components. These are the chemical potential, the difference in protons, and the electron potential due to the electrostatic charge of the protons. Fermentation is the aerobic method of cellular respiration, as it does not. Fermentation is the anaerobic method of cellular respiration, as it does not require oxygen. The two types of fermentation are lactic acid and alcoholic fermentation. In lactic acid fermentation, the electron receptor is pyruvate, and the products are 2-lactate and 2-ATP. While in alcoholic fermentation, the electron acceptor is still pyruvate, but the products are 2-ethanol, 2-CO2, and 2-ATP. There are three ways to regulate metabolic pathways. Firstly, gene expression can be regulated to control the amount of active enzyme. Secondly, enzyme activity can be changed by covalent modifications. And thirdly, there can be feedback inhibition from allosteric enzymes. Light is a form of energy, but it is specifically electromagnetic radiation, which means it is propagated as waves and as particles. Photons are packets of energy. Absorbed energy boosts an electron in a molecule to a shell further from the nucleus. Pigments absorb specific wavelengths of the sun's light. Not all wavelengths, however, can be absorbed, so some are deflected. The deflected wavelengths determine how we will see whatever thing reflects it. As an example, chlorophyll reflects green wavelengths, which is why we see plants as green. Photosynthesis is very important as it is the method that plants get the energy they need. It also releases oxygen as a byproduct, which is obviously very important for humans. Stomata are pores on the surface of leaves where CO2 enters and O2 and H2O leave. The photosynthesis reaction is 6CO2 plus H2O and light energy creates glucose and 6O2. The two pathways of photosynthesis are light reactions and light-independent /carbon reaction /Calvin cycle. The reaction converts light energy to ATP and NADPH, and the light-independent reactions use the ATP and NADPH from the light reaction and uses those plus the CO2 to produce carbohydrates. Photosynthesis is a multi-protein light harvesting the photosystem is a multi-protein light harvesting complex in the thylakoid. These photosystems are used in the light reaction pathways of photosynthesis. Light harvesting complexes are composed of chlorophyll and accessory pigments. These, of course, absorb the light energy. The reaction centers are where redox reactions occur that convert the light energy into chemical energy. The final electron receptor in the reaction center is NADP+. Non-cyclic electron transport utilizes both photosystem 1 and 2, while cyclic only uses photosystem 1. Non-cyclic produces ATP and NADPH, while cyclic produces ATP instead of NADPH. The way the ATP is formed in photosynthesis is quite similar to the cellular respiration, with protons being transferred across the thylakoid membrane to the lumen, creating an electrochemical gradient, and then the high level of proteins in the lumen forces the protons back into the stroma through ATP synthase. ATP synthase is of course vital as it is very important to multiple methods of creating ATP. In light-independent reactions, CO2 is reduced to carbohydrates. The energy in ADP and NADPH from the light reactions is used to reduce CO2. This occurs in the stroma. The enzyme that catalyzes the reaction is the Calvin site is the wait, what? Hold on. The enzyme that catalyzes the reaction in the Calvin cycle is Rubisco. For each turn of the Calvin cycle, CO2 is fixed, one CO2 is fixed, and one RuBP is regenerated. Photorespiration is where RuBP reacts with O2 instead of CO2. This is not good. C4 plants attempt to combat photorespiration by spatially separating the initial CO2 fixation in the Calvin cycle, while CAM plants attempt to combat it by only opening stomata at nighttime. Some examples of energy loss during photosynthesis include not all wavelengths of light being usable, loss due to heat, energy loss in the electron transport chain, and photorespiration, if that one counts. And Exam 4 Study Guide. 4 events of cell division. Firstly, the reproductive signal is signaled, and it initiates cell division. It can be intracellular or extracellular. Next is the replication of DNA. Third is segregation, the separation of DNA into the two cells. And then fourth is cytokinesis, which is the separation of cell materials into the two cells. Prokaryotic cell division is called binary fission. It takes place in the 4 steps listed above, those being the reproductive signal, replication of DNA, segregation, and cytokinesis. A prokaryotic cell will divide when triggered by an extracellular signal, such as environmental conditions. Then it will replicate its DNA. This occurs through a replication complex, beginning at the ori and ending at the ter, which are the origin and terminus, basically, the beginning and the end, to remember it easily. After this, the segregation occurs by the ori regions moving to opposite ends of the cell, and then in cytokinesis, the membrane is pinched in, and the protein fibers form a ring. The difference between eukaryotic cell division and prokaryotic in terms of when it occurs is that prokaryotic cells will constantly divide so long as conditions are correct, while eukaryotic cells are part of a greater organism and only divide when a signal from elsewhere in the organism tells them to. Sister chromatids are recently replicated chromosomes that are still closely associated with each other. Mitosis is the process that separates 2 recently replicated chromosomes into 2 new nuclei. The cell cycle has 3 broad phases. These are interphase, mitosis, and cytokinesis. Interphase has 3 subphases. These are G1, S, and G2. The first of these is G1 phase, which is the first where the cell has not yet begun the process. Once it enters S phase, the DNA replicates and the sister chromatids stay together. And in G2, the cell prepares for mitosis. To enter S phase, however, the cell needs to pass the restriction point. The restriction point is what prevents cells from constantly dividing. The factors involved in passing the restriction point are cyclin, CDK, and RB. Cyclin is an allosteric regulator, a type of enzyme, which binds to CDK when it needs to be activated. CDK is cyclin-dependent kinase, which once is activated can be used to phosphorylate RB. RB is the retinoblastoma protein. Normally, it is active and inhibits the cell cycle. However, it is deactivated by CDK phosphorylating it, allowing the cell cycle to occur. G0 phase cells are cells that are not in the cell cycle. To be activated, they must be stimulated by growth factors. These growth factors will activate pathways that end with cyclin production. There is a lot of DNA, so it needs to be packed together by proteins called histones. Nucleosomes are bead-like structures that are formed as a result of this interaction between DNA and histones. A chromatin is a DNA module bound by proteins. A chromosome is a long, thread-like structure made up of DNA and proteins that contains genetic information. If the chromosome is in a X shape, that means it is the metaphase of mitosis. Cohesins are proteins that hold sister chromatins together during G2. The centrosome is the region of the chromosome that acts as a site for sister chromosomes to connect. Also, it is the middle of the chromosome, so it's the little kind of, most pictures, the dot in the middle that they all connect to. The first phase of mitosis is prophase. In this phase, the chromosomes condense, the spindles form, and the centrosomes move to opposite sides of the cell, and in late prophase, kinetochores form, which are vital for movement of the chromosomes. Then there is prometaphase, in which the nuclear envelope is broken down. All cohesins are removed, except for the centromere, and the chromosomes attach to the spindles. Then is metaphase. Then the chromosomes align themselves along the middle of the cell, called the equatorial plate. Then anaphase occurs. This causes the separation of the chromosomes. They are referred to as daughter chromosomes after the separation. These daughter chromosomes begin migrating to the poles, opposite ends of the cell, and following this is telophase, in which the nuclear envelopes and nuclei reform and the chromatin decondense. APC is important as it hydrolyzes cohesin, allowing the chromosomes to separate. In animal cytokinesis, proteins cause the pinching of the cell membrane that splits the cell in two, while in plant cytokinesis, vesicles appear along the plane of cell division, creating new plasma membrane. A gamete is a sex cell created by meiosis. Somatic cells are body cells not specialized for reproduction. Homologous pairs are matching pairs of chromosomes that carry the same genes for the same traits. Haploid means half the chromosomes, 23, while diploid is twice as much, 46. Fertilization is the combination of two haploid gametes to form a diploid zygote. A zygote is created by two gametes, also known as a fertilized egg. Zygotes grow through mitosis, not meiosis. Cells that undergo meiosis are specialized cells called germ cells. In males, they produce sperm cells, and in females, they produce egg cells. Meiosis occurs in two broad steps, meiosis 1 and meiosis 2. The first step of meiosis 1 is prophase 1, in which the chromatin begins to condense. The next part of prophase 1 is prometaphase, where crossing over occurs. This is where non-sister chromosomes exchange genetic material, giving more varied genetic results. Next, in metaphase 1, homologous pairs line up on the equatorial plate. This is different than mitosis, as it is the pairs line up instead of just the chromosomes. Then, in anaphase 1, the homologous chromosomes, each with two chromatids, move to opposite sides of the cell. And in telophase, the chromosomes gather into nuclei, and the original cell divides. Then meiosis 2 begins with prophase 2. In prophase 2, the chromosomes in the two sister cells condense but do not replicate. Then in metaphase 2, the centromeres, remember the middle of the chromatids, line up along the equatorial plate. These are then pulled apart into chromosomes in anaphase 2. Each new cell has a unique genetic makeup due to crossing over and random assortment. And finally, in telophase 2, the chromosomes gather into nuclei and the cells divide, resulting in 4 daughter cells. And each daughter cell has a haploid number of chromosomes. Non-disjunction is an error that can occur during meiosis. This occurs when homologous pairs during anaphase 1 or sister chromatins in meiosis 2 do not separate correctly. This can result in too few or too many chromosomes. In humans, having an extra chromosome 21 causes Down syndrome. Mitosis and meiosis are both processes of cell division, but they differ significantly in purpose and outcome. Mitosis is used for growth, repair, and asexual reproduction, producing 2 genetically identical diploid daughter cells. It involves 1 round of division and does not include crossing over. In context, meiosis is used for sexual reproduction, producing 4 genetically diverse haploid gametes, sperm or eggs, through 2 rounds of division. Meiosis includes crossing over during prophase 1, which leads to genetic recombination. Mitosis produces somatic cells, while meiosis produces gametes, and the latter is essential for maintaining genetic diversity in offspring. Apoptosis is programmed cell death. It is important as old and damaged cells can cause damage to the body. Signals to initiate can include hormones, growth factors, viral infections, toxins, extensive DNA damage. A tumor is a large mass of cells. Benign tumors grow slowly, resemble the surrounding tissue, and remain localized, while malignant tumors do not resemble the parent tissue, and assumably also do not remain localized. A gene is DNA that encodes info to build proteins. Gregor Mendel is the father of genetics. The phenotype refers to the physical appearance of an organism, while the genotype, is the genetic makeup of the organism. Dominant alleles always express their proteins, while recessive don't whenever a dominant is present. Homozygous refers to having 2 of the same alleles, either dominant or recessive, while heterozygous refers to having 1 of each. There is no such thing as heterozygous dominant or heterozygous recessive, they just have 1 of each. P refers to the parent generation, or the first generation of plants Mendel worked with. F1 then refers to the first generation of offspring, and F2 to the next generation of offspring after that. Offspring inherit alleles from both the mother and the father. Gregor Mendel had 2 laws. There is the law of segregation. The 2 copies of the gene separate during gamete formation, and each gamete receives only 1 copy. And the law of independent assortment. Copies of different genes assort independently. Punnett squares are easy. Remember how to do them. Line up the set of genes on top, and line up 1 set of genes on top, 1 on the bottom. 3-to-1 ratio for 2 heterozygous, the 1-to-1 ratio for a heterozygous and a homozygous recessive, 9-3-3-1 for 2 double heterozygous. You got this, it's easy. Remember the 2 most simple statistic rules, the multiplication and addition rules. This is brain dead easy. If you fail this on the test, you're dumb. Don't screw it up. It's very easy. The wild type is the allele present in most of the population. A mutation is a stable inherited change in genetic material. Incomplete dominance refers to when alleles are neither dominant or recessive, while codominance occurs when more than 1 allele encodes a functioning protein. A locus is a specific point on a chromosome. Pleiotropic refers to 1 allele with multiple phenotypic effects, and epistatus refers to the expression of 1 gene being influenced by another gene. Linked genes are genes that are close together on the same chromosome, which usually travel together during crossing over. Light, temperature, and nutrition can all affect the expression of the genotype. Siamese cats are a great example of this, as their extremities are darker in color because the enzyme that produces the dark color is more productive in low temperatures than high temperatures. And obviously the extremities are at a colder temperature than the rest of the body. And prokaryotic cells exchange cells through conjugation. Plasmids are circular chromosomes in prokaryotes. These plasmids contain genes for unusual metabolic processes, antibiotic functions, and for making a sex pilus, whatever that is. Alright, and there you go. Alright. Study guide for DNA, RNA, and evolution topics. DNA. The importance of Franklin, Crick, and Watson. Franklin's X-ray crystallography revealed DNA's helical structure, aiding Watson and Crick in proposing the double helix model. Structure. The key features. 1. The double helix structure. 2. Sugar-phosphate backbone. 3. Nitrogenous base pairs. And 4. Anti-parallel strands. Components are sugar, phosphate group, and nitrogenous bases, which are the A, T, G, and C. Location is they are found in the cell nucleus, also in mitochondria and chloroplasts. Pairing for DNA and DNA/DNA, so that doesn't really make a lot of sense, but we'll just go with it. DNA/DNA, A's pair with T's, G's pair with C's. For DNA RNA, which makes a little more sense, A's pair with U's and G's pair with C's. Chargaff's rule is that the percentage of A's equals the percentage of T's, and percentage of G's equals the percentage of C's. DNA replication. Definition: process of copying DNA before cell division. Semi-conservative. Each new DNA molecule has one original and one new strand. Steps of DNA replication. 1. Initiation. Begins at the origin of replication. Helicase unwinds DNA and replication forks. 2. Elongation. DNA polymerase adds nucleotides in the 5' to 3' direction. Leading versus lagging strand. Leading strand is synthesized continuously. Lagging strand forms short Okazaki fragments that are later joined by DNA ligase. Key enzymes and structures. DNA polymerase synthesizes new DNA strands. DNA helicase unwinds the DNA helix. DNA ligase seals gaps between the Okazaki fragments. And primers, short RNA sequences that initiate DNA synthesis. DNA repair mechanisms. 1. Mismatch repair fixes incorrect base pairings. 2. Base excision repair removes damaged bases. And 3. Nucleotide excision repair, which repairs larger helix distorting lesions. LESIONS. Sure. Polymerase chain reaction, which is PCR. Definition: a method to apply specific DNA sequences in vitro. Gene expression. There are 2 steps. 1. Transcription. DNA sequence is copied into RNA. And 2. Translation. The RNA sequence is used to synthesize a polypeptide. Central dogma. DNA codes RNA, codes protein, proposed by Watson and Crick. Exception: reverse transcription in retroviruses. RNA. There are 3 main types. Messenger RNA, which is mRNA, carries genetic code from DNA. Ribosomal RNA, which is rRNA, catalyzes peptide bond formations. And transfer RNA, which is tRNA, brings amino acids to ribosomes. The transcription has 3 steps. First is initiation. RNA polymerase binds to the promoter region. 2. Elongation. RNA polymerase synthesizes RNA in the 5' to 3' direction. And 3. Termination. RNA synthesis stops at a terminator sequence. Promoters. DNA sequences that signal the start of transcription. Transcription factors. Proteins that assist RNA polymerase binding. Differences in prokaryotic and eukaryotic gene expression. Introns, non-coding, are spliced out of eukaryotic pre-mRNA, leaving exons, coding regions. Pre-mRNA processing in eukaryotes includes the addition of a 5' cap and a poly A tail. Genetic code and translation. Genetic code specifies which amino acids are encoded by codons, which are the 3 base sequences. A start codon, AUG methionine, and a stop codon is a UAA, a UAG, or a UGA. Those are going to be a pain in the neck to remember, aren't they? UAA, I'm sure there's something that would make that memorable, and then, well, UGA is that like the University of Georgia? I don't really remember. But then there's also UAG. So UAA, UAG, and UGA. So it's like the G is sneaking up. It's slowly crawling across. It starts completely off, then takes over the first A, then takes over the second A. But that slides that first A down to that second A spot. So UAA, UAG, UGA. Steps of translation. First is initiation. The ribosomes assemble around the start codon. Second is elongation. Amino acids are added to the polypeptide chain. And third is termination. Translation stops at a stop codon. Evolution and natural selection, which is near the end. Evolution is change in allele frequencies in a population over time. A key figure of this is Charles Darwin, who proposed natural selection and On the Origin of Species. Natural versus artificial selection. Natural selection is driven by environmental pressures, while artificial selection is human-directed. Modes of natural selection are stabilizing, directional, and disruptive. Hardy-Weinberg equilibrium. Definition: describes populations where allele frequencies do not change. Assumptions are no mutation, random mating, no natural selection, large population size, and no gene flow. Equations that we need to know are that p squared plus 2pq plus q squared equals 1, and p plus q equals 1, where p and q are allele frequencies, and then the p squared and q squared are the population of homozygous dominant, and the q squared is the homozygous recessive, and then the 2pq is for heterozygous. So it's a pretty simple equation there. Genetic drift. The definition is random changes in allele frequencies. Examples of this includes the founder effect, which is small group establishes a new population, and then the bottleneck effect, which is population size is drastically reduced. And okay, that's it. Good job, you listened to this whole hour-long video. Now listen to it again, you fool. You are gonna hate me saying this by the end, but you're gonna listen to it again and you're gonna like it, okay?